and emission factors. And you can see the main emission calculation formula. Emissions of PCDD in air, residues, and land, as well as PCBs and HCBs in air, were prepared. PCDDs and PCBs emission were expressed in milligram toxic equivalent and HCB in milligram. Emission at the preliminary stage of study, the main sources of POPs releases in Antarctica were identified. The list of POPs emission sources in comparison with other continents is short because anthropogenic activity is very limited and mostly connected with scientific station operation and maintenance. Taking into account the international and national experience of unintentional POPs inventory, fuel combustion by stationary and mobile sources, as well as waste incineration, were considered as main sources of POPs emission for numerical evaluation. Shipping and aviation are not significant sources of POPs emission at regional and national scale, and these sources were counted semi-quantitatively only. Due to Antarctica's specific status, there is no system of statistics collection, possibly except for tourism. Therefore, there is no aggregated human activity data per sector for the whole continent, which is necessary as input data in POPs emission calculation. That is why a combination of approaches was applied to derive activity rates for Antarctica. For modern period, available records on stations and national Antarctic programs were used, including volumes of fuel and waste burned, types and capacity of power installations, number of vehicles, station population, etc. For this purpose, different sources of information were taken into account, including annual reports, results of inspections, initial and comprehensive environmental evaluation, data of COMNAP, scientific publication, and so on. For the late 1980s, previous estimates of fuel consumption in Antarctica by stationary and land-based mobile sources were used. Estimation of the volume of waste burning was made taking into account the published data. All types of data were analyzed and prepared in required format. Some assumptions were made in case of data absence or incompleteness. As a result, the total fuel combustion in Antarctica, as well as fuel combustion by sector, including energy production, heating systems, mobile sources, were assessed. And the volume of waste incineration was also assessed. As I mentioned before, modern period and the late 1980s were taken into account. Speaking about emission factors, it should be stressed that POPs have not been measured in waste gases in Antarctica. Therefore, possibilities to justify the selection of emission factors were very limited. In this study, emission factors given in the toolkit for identification and quantification of releases of dioxins, furans, and other unintentional POPs prepared under Stockholm Convention on POPs were used. Some additions to toolkit was also used. You can see the emission factors for different sectors and for different chemical substances. As main result is presented on this slide, and you can see annual POPs emission from different anthropogenic sources in Antarctica for modern period. According to estimates, PCDD's air emission comprises about 60 milligram toxic equivalent in air and about 2.5 milligram toxic equivalent are emitted annually in residues. PCB's air emission comprises about 5 milligram toxic equivalent and HCB's about 457 milligram. Waste incineration makes the greatest contribution in the total PCDD emission. It makes about 96% of the total air emission. There is a similar situation for PCBs. Waste incineration makes up 98% of the total PCBs emission. Concerning HCB, there are two main sources of emission in the environment: ground vehicles and waste incineration, which contribute about 90% of the total HCB's emission. Annual PCDD emission for the late 1980s amounted about 770 milligram toxic equivalent in air and 25 milligram toxic equivalent in the land. Burning waste, mostly open, was the major sources of POPs emission for that time. This is evidence of environmental effects of such practices for a long time. After the protocol of environmental protection had been signed and had entered into force, construction of incinerators at different stations and implementation of other measures were undertaken. To understand how much POPs are emitted annually in environment of Antarctica, comparison with emissions of gateway countries was fulfilled. I mean, Argentina, Chile, South Africa, Australia, and New Zealand. Gateway countries were selected due to their close location to Antarctica and possibility of greatest direct air impact. Besides, these countries are active in Antarctica. National reports to the Stockholm Convention, I mean 4 rounds, were used as sources of POPs emission data. Comparison of dioxin furans emission and in Antarctica and in gateway countries was made. And as expected, the values of dioxin furans in Antarctica in comparison with other countries is very low. Currently, it is in the range 0.008 to 0.2% of emissions in particular country. However, the calculation of PCDD annual air emission per person in Antarctica, taking into account the maximum population at the station, has proved that specific PCDD emission value in Antarctica is in the range of this value in other countries. And historical emission exceeds its values significantly. To conclude the presentation, I would like to underline that retrospective assessment shows that our 30-year period air emission of PCDD and other unintentional POPs decreased significantly. And this is due to prohibition of open burning of waste and reduction of waste burning. Speaking about further task, I would like to say that improved account of waste incineration practices and incinerated volumes, as well as direct measurements of POPs in flue gases are very important. Further measures for improving incineration practices, the use of afterburns, dust and gas collection equipment is necessary. And it is very important also establishment of emission reporting in the framework of Antarctic Treaty System. It will be very useful for POPs management in Antarctica. Thank you very much for your attention.
Thank you very much, Tamara, for that excellent presentation on a really important area of knowledge gap. So do we have any questions from the audience for Tamara? We have a few minutes before our next speaker. If not, I am...
Sorry, no, I can't see any question.
Okay, I'll jump in with one. So Tamara, the data, I know I'm sort of aware that this has been a concern for a number of years amongst particularly the COMNAP community, and there has been a push for better incineration practices or elimination of incineration practices. I'm just wondering where the data for your calculations, as in the types of incineration practices implemented by different stations, is there a good catalogue of that available? And where did you source that information from to make the calculations?
Thank you for your question, Susan. Not so well data, yes, only, as I remember, only the data available for 16 stations, and we used the different other data regarding the types of incinerators and volumes of waste burning, and we calculated the volume of incineration per capita, per person, taking into account the incinerated volume of waste and the number of persons per station, and then translated this data for other stations.
Yes.
Yeah.
I see. Oh, good. No, I very much enjoyed reading the paper because it has been an overlooked aspect of legacy contaminant pollution in the Antarctic. So well done on that presentation, Tamara. If there are other questions, we might leave it till the discussion at the end, and we'll move on to our next presenter, Andrea.
Okay. Everyone can see my presentation?
Yes, just put in
Yes.
presentation mode. Good.
Okay.
Perfect.
Hello, everyone, or good morning, or good afternoon, or good evening. I'm here to present my research work about perfluoroalkylated substances, an investigation of oceanic transport across the Antarctic circumpolar current. First of all, PFAS are persistent organic pollutants. But what are persistent organic pollutants? We used to categorize these chemical compounds under the Stockholm Convention, and they have to own precise chemical and physical characteristics to be considered in this convention, like environmental persistence, the capacity for long-range environmental transport, the ability to bioaccumulate in the organisms, and they have to be toxic. Traditionally, many of the legacy POPs behave predictably in base of their chemical characteristic. That means they are highly volatile. Thanks for these characteristics, they can move from the low latitude or mid latitude to the high latitude in two different ways. The long range atmospheric transport, where compounds highly volatile can evaporate in warm regions and travel for long distance before condensing in colder region. And in another way, the shorter range atmospheric transport for the less volatile compounds and consists of a series of cycles of evaporation and condensation in a process known as grasshopper effect. The final destiny for these both paths for the chemical compounds is to be trapped in the colder region, like, for example, Antarctica in this case. But why this introduction? This introduction about the behavior of legacy POPs, it's because PFAS, or perfluorinated alkyl substances, changed our view about how they can move in the environment, mainly because of their chemical properties. PFAS are obviously man-made chemicals that have been in use since the 50s, and they had added to the Stockholm Convention since 2009. Speaking of chemical characteristics, they contain a chain of carbon atoms bonded to fluorine atoms, and some compounds also have a functional group at the end of the chain. This structure leads them to have amphiphilic properties and make them largely used in a lot of products for industries or consumers. For example, firefighting foams, fabrics, the leather manufacturing process, paper or carpet. And these are only few examples of products that we can find in our home where PFAS are present. These amphiphilic properties change the way they can move, as I mentioned before, in the environment. Even if it's not yet totally clear how they can move in the environment, there are two main hypotheses about this topic. The first is the atmospheric transport, where it is important to consider the precursors of these chemical compounds. For example, the fluorotelomere alcohols. There is a volatile compounds that can biodegrade in perfluorinated carboxylic acid in elsewhere, a far distance from the point of origin, or we need to consider the marine aerosol, where PFAS are present, and they can be mobilized in the atmosphere again after an event, and maybe this kind of transport, the atmospheric transport, can explain the presence of these compounds in, for example, snow deposition or glacier in the cold environment. Another way they can move is across the ocean and the marine currents. Sorry. Speaking, an important characteristic to consider in this transport is that the ionic form of PFAS demonstrate low volatility and so are less prone to a long-range atmospheric transport. But they show to be hydrophilic, so they can dissolve in water and accumulate in the world surface water. In these last years, the oceanic transport has been proposed as a principal mechanism for this dispersal. Following the ocean currents, PFAS can spread widely, reaching even very remote areas from the point of origin, like in this case, we are considering Antarctica. But speaking about Antarctica, the Antarctic circumpolar current play a key role in this environment. In fact, it's the largest and strongest current present around the continent, and it presents a physical barrier for effective north to south transport of PFAS into the Antarctic waters. However, the barrier is only temporary, because some exchange of deep upwelling waters occurring into south of this circumpolar current. What is the main of this research? The aim of this study is to understand whether the ionic form was able to move over long distances in surface water, because it's well known the presence of PFAS in water South Pacific, but not in Southern Ocean surface water. And moreover, the presence of PFAS in the water surrounding the Antarctica continent is not well documented. To fulfill this lack of information, it was designed a sampling campaign where water samples were collected along the longitudinal sampling transect from Hobart to the Antarctic continent in 2013-2014. The samples were collected every degree of change in latitude, 2 liters per sample, and every half degree in the polar front. With the polar front, we mean the boundary between the polar cell and the Ferrel cell around the 60th grade in latitude. The samples were collected through the uncontaminated seawater line and were accompanied by 5 to 7 field blanks of pre-extracted milky water. Once the sample was collected, were sent to Aarhus University for the analysis, and the analysis consisted to the extraction of the water sample 1 liter at a time through wax cartridge, and once the extraction happened, the cartridge was washed with ammonium acetate buffer and dried, then eluted with methanol, and the extract was reduced to dryness with gas nitrogen. After this, the sample was reconstituted in methanol and analyzed by liquid chromatography tandem mass spectrometry with the electrospray ionization in negative mode, just to have a little information about the method. We know now hundreds of compounds in PFAS categories, but for this study, the compounds were considered the compounds considered were these, divided in two categories, PFSA, perfluorosulfonic acid, and PFCA, perfluoroalkyl carboxylic acid. The expectation in this result for this study, it was to see a decrease of contaminants from before the polar front to the after the polar front. This is the results about PFOS, the compounds in the category of perfluorosulfonic acid. And in these categories, PFOS was the mainly presence in the compounds analyzed. In the graph, we can see in the x-axis the location, that means the latitude where the sample was collected, and in the y-axis the concentration in nanogram per liter. The dot line represented the polar front. It is possible to notice that the concentration of PFOS, in this case, decrease after the polar front, even if his presence is consistent and is important even after the polar front. But this result is consistent with some previous studies, because PFOS is a widely large presence all around the world. Considering the remaining part of the compounds in the perfluorosulfonic acid category, we can see that for some of the compounds there is a decreasing of the concentration of these chemicals, but for example, for the perfluorohexane sulfonate, the orange line, there is an increasing concentration. If you be patient, the next slide I can show you the PFCA perfluoroalkyl carboxylic acid results, and then I'm gonna explain another interesting point about the perfluorosulfonic acid. We here can see exactly what we expected. That means the dramatic decrease of the contamination in after the polar front. To confront with the previous graph, the conclusion that we can reach in this study are these. During this investigation, it was possible to verify the contamination of the waters south of the Antarctic circumpolar current is already taking place. And another possible explanation about the increasing of certain compounds after the circumpolar current is the presence of the research station located on the continent as a direct source of contaminants in the seawater around the continent. It was a consideration. It's important to consider this because in the Antarctica station there are a lot of products with PFAS, and the contamination can be happen directly in the continent to the surrounding waters. Another important thing to considering after this study is the timing, how long the contaminants can use to move through the ocean to the Antarctica station. Thanks to these authors, Speech et al. And Udicon et al., we can say that PFAS, for example, PFOS and PFOA, can move from 50 to 700 years from the deposited from the origin source in the Western USA ocean in sea to arrive in Antarctica.
That's
This
20 minutes.
information, sorry?
That's your 20-minute mark. Sorry, your.
Yes, I finished. This is important because the peak of the usage of the PFAS it was in 80s, and we can expect an initial input in 2030, a massive initial input because of this timing. My future perspective are to collect new water samples in the transect from South America to Antarctic Peninsula and compare the PFAS concentration between Western sector and the Eastern sector to better understand the spatial distribution with respect to the Antarctic circumpolar current. Thank you very much.
Excellent, Andrea. Thank you very much for that presentation. I'll open up the floor to any questions.
Sorry, during the presentation, I can't see the question box. If Catherine can help.
Yeah, no, there's nothing there at this stage. And apologies,
Okay.
that was your 15-minute warning. So we should say that, Susan, that we're 15 minutes, not warning, just an alert, just so you know you have to wrap up, if that's okay for speakers.
Sure.
Yeah, it was perfectly okay. Don't worry.
Right. Sorry about that. Thank you.
Thank you.
No problem. Okay. So do we have any questions for Andrea regarding PFAS in the Southern Ocean? If not, thank you very much again, Andrea, and we'll shift the presentations over to our next presenter, who is Professor Nicoletta Adamolo from the Institute of Polar Sciences, National Research Council of Italy, and she'll be presenting to us the influence of climate change on bioaccumulation patterns of legacy and emerging POPs in Antarctica. Welcome, Nicoletta.
Thank you, Susan. Sorry. I'll start from the end. No worries. So, everything is okay? You can see.
Very quiet from my end. Nicoletta, very quiet.
Very quiet? Oh, sorry. Now it's better now?
A little bit, yes. Slides are good. Yep.
Okay. So this talk this morning for me is about the influence of climate change and POPs, the behavior of POPs in the Antarctic ecosystem, also from a bipolar perspective. So first, I want to summarize a few key information on the Antarctic ecosystem, the human impacts and climate change. The human impact on Antarctica and the Southern Ocean are still limited due to the remoteness and extreme climate. The continent is really a cold desert, and also the seawater temperature is a stable temperature about -2 degrees centigrade. The presence of POPs in the Antarctic continent and the Southern Ocean is mainly due to the long-range atmospheric transport. In fact, the Antarctic circumpolar current acts as a natural northern boundary between the cold sea waters of the Southern Ocean and the warm sea waters of the other oceans. So these features are supposed to cause a delay in the POP transport and bioaccumulation when compared to the Arctic. The Antarctic marine ecosystem are also very fragile, meaning that even small variation can cause extreme changes. They also show low resilience with respect to other ecosystems. In this context, the climate crisis can have a devastating impact on this ecosystem, including effects on the food web structure and the availability of food resources. These perturbations may shift the trophic position of organisms in their trophic webs and consequently the pattern of pop bioaccumulation. The climate change occurs also in this region. So for now, it was reported that the Earth's surface temperature has increased from 3 to 7 degrees centigrade in the 50s, mostly in the West Antarctica. However, scale, since East Antarctica is colder and these events have been rare so far. So this review summarizes a section of the report titled POPs and Chemicals of Emerging Arctic Concern, Influence of Climate Change, which is an assessment of the Arctic Monitoring Assessment Program. The comparison between observation reports in the Arctic and the Antarctic region was included in the AMAP assessment because it can provide a global perspective on the effects of climate change on contaminant trends in polar regions that have different remote sources and transport pathways of contaminants. So the presence of POPs in the Antarctic ecosystems has been reported in an increasing number of articles since the 60s, but the number of these articles are still very low compared with the number of the papers published in the Arctic environment. So the articles on the presence of POPs, together the time series and correlation to climate parameters are lacking for Antarctic environments. Thus, we consider for this paper, this assessment, peer review articles that link contaminant concentration and biological parameters. We select articles on marine fishes and adult penguins because these species are native to Antarctic marine environment and thus suitable to study temporal change of contaminant exposure. The terrestrial ecosystem is cover only 0.34% of the continent, currently without ice, seasonally or permanently. This area are colored in red in the image on the right. In these ice-free areas, glaciers, freshwaters, lakes, and ponds can be present as shown in this picture. The picture on your left was taken at Monsoon Point near the myriad station, a seasonally ice-free area in the Victoria Land. And you can see the park, a lake, and a small pond. The other picture was taken on the dry valleys with some glaciers. So the comparison in the assessment of the temporal trend in the Arctic terrestrial environment is focused on freshwater fish, mostly from Greenland. So the comparison to the Antarctic freshwater environment is not really possible. Therefore, there is an urgent need to intensify the research in these environments. In fact, many scientific stations are located in these free ice areas, which may be more vulnerable to human impact. To give you an idea of the POP concentration, PCBs and PBDA in the Victoria Land are very low and among the lowest worldwide. In these environments, local sources of PBDs include the scientific station, field camps also, and seabird colonies. The lakes and ponds in the Victoria Land in this article in this very important study. These authors suggested that the remobilization of PCBs is driven by change in temperature and soil content of organic matter. They suggested that the current and future POP remobilization and things in Antarctica, besides warming, are a function of the close coupling of climate change and changing carbon cycling. Moving to the marine ecosystem, there is a study on the pop temporal trends on the emerald rock cod, the Trematomus bernacchi, from the Ross Sea that covered a period of 30 years, from the early 1918 to 2011. These authors observe a generally decreasing trend of pops with two elevated concentration peaks in 2001 and 2005. These peaks were ascribed to the calving of the iceberg B15 from the Ross Ice shelf in 2002. This event may have caused the release of pops, which were rapidly mint, into the seawater. Once in the water column, pops can bioaccumulate in the lower trophic levels and then up to the predators like seabirds. So, the decreasing concentration of pops observed in the Antarctic marine organism over time generally correspond with declining trends in seabirds. A study reports the pop temporal trends in resident and migrating seabirds, the Adelie penguin and the South Polar skua. In the Adelie penguin, there is an evident trend for PCB concentration since the 90s, but this trend is not equally clear for the DDTs, increasing again in the first decade of 2000. The South Polar skua is a migrating seabird that nests along the Antarctic coast, mostly near penguin colonies. Skuas feed on their eggs and chicks, and it is interesting to note that a temporal trend cannot be described for this species. Maybe its migrating habits may alter the bioaccumulation trends since skuas overwinter in more polluted northern areas. This interesting article explored the potential effects of climate change on DDT levels in Adelie penguins nesting in three different areas of Antarctica: the Antarctic Peninsula, the Ross Sea, and the eastern coast. The authors reviewed some articles and report the results in this graph. On your left, the graphs show the predicted levels agree with measured values. In the graph on your right, the ratio DDE significantly decreased from the 60 to 2010. So the concentration of DDTs in penguins did not decrease from the 70s to the first decade of 2000, which is in contrast to the observational report for seabirds in the Arctic during the same period, and in agreement also with previous studies on seabirds. So to conclude, the temporal trends cannot be evaluated and compared to the Arctic in the freshwater environment, since we have no fish in the freshwater environment in Antarctica. So we are really a lack of data for these temporal trends. Also in the marine environment, the lack of records of climate parameters hinder the assessment of temporal trends in comparison to the Arctic. However, pop concentration has declined over the last decades in some Antarctic fish, but these temporal trends appear to be weak and inconstant. The concentration peaks of pops observed in Antarctic fish were potentially related to their release after an iceberg calving in the Ross Ice shelf. These effects have not been studied in Arctic to any significant degree, but are likely to occur given the similarities in polar environments. So further studies should include the recording of climate parameters and the biological parameters also of the organism in correlation to pop concentration and bioaccumulation. The systematic monitoring of target species in marine ecosystem and of abiotic samples like lichens and moss in terrestrial ecosystems. Moreover, this new data should be compared to the results from retrospective studies. The peculiarity of the marine Antarctic ecosystem, the effect of the pelagic benthic coupling and the contaminant profiles and trends leaves still open the question whether the pop amount accumulating in the Antarctic ecosystem is really decreasing or not, depending on the climate-driven change, since concentration peaks can still be observed in biota. So this is my last slide, and thank you for your attention.
Thank you very much, Nicoletta, for that excellent overview. I very much enjoyed digging into some of those gaps that still exist for us in the Antarctic, and revisiting some of the important papers on the topic over the years. Do we have any questions for Nicoletta from the audience?
No. There aren't there aren't questions from the audience.
Okay. Well, I might ask one. So, from your work, and Nicoletta, with the AMAP reports and getting that bipolar perspective, what would be your recommendation for longer term monitoring that in the Antarctic for biota? What would you suggest? You had some species there that already have somewhat of a timeline. Are there any that stand out to you as being better than others, or ones that we should be focusing our attention on?
I think it's really important to continue to study the resident and migrating seabirds to understand also the importance of the pathways of contamination that the migrating organism and animals may arrive in Antarctica, and also really start with systematic sampling and with the biota, together with the climate parameters, the meteorological parameters like snow cover, humidity, temperature, wind direction, and also really important the biological parameters of the biota sample. So I think we can start to correlate more and more these three areas. So chemical, ecotoxicological, biological, and climatic. Yeah.
Excellent. Yeah, I think that's absolutely correct to elucidate some clarity on what parameters, what's important to measure. So monitoring what matters, so to speak. Do we have any other questions? We're slightly ahead of time here, so we do have time to discuss further before our next presentation. So the data on the Adelie penguins that you presented, Nicoletta, is that all from the Italian programs that you're involved in?
No, not only. Yes, most of all, we have our papers with Simonetta, Corsolini. No, there is also the last paper I present is a review with the 3 areas of Antarctica. So it's really a good, and there is a lot of data also from the French station also and the peninsula. Yeah.
Mm.
But we are focusing this review also with the papers that put together the climatic parameters and the biological data of the organism. So the paper really decrease in numbers when you look.
Yeah, that's right. I guess you've run into the problems that we often do, where we haven't with small sample sizes and different regions of the continent. So that's certainly, an area that we have that you've also highlighted here, that we need for more systematic and continuous monitoring across the continent. So it was a very lovely overview. If we don't have any more questions for Nicoletta now, we might move ahead. So our next presenter unfortunately had to withdraw, Phoebe Lewis from RMIT, who has also worked closely with the Australian Antarctic Division through her PhD, has actually come down with COVID over the past week. So she's had to unfortunately withdraw her presentation. So we are moving our last two presenters up in the schedule, which means that our next presenter, Omolara Omomo from, again, my team at Griffith University, Southern Ocean Persistent Organic Pollutants Program, will be presenting her honours research, which outlines the first comparison of humpback whale phthalate burdens in high and low pollution areas. So, Lara, if you'd like to bring forward your presentation, please.
Sorry, just give me a moment. I'll just set up quickly. So the first comparison of humpback whale phthalate burdens in high and low pollution areas. So phthalates are additives in various consumer products, including fertilizers, personal care products, fragrances, and pesticides. However, the most common use is as plasticizers in plastic products. So plasticizers, they increase the flexibility of plastic and they sit within the plastic matrix. However, they are not covalently bound to this matrix, so they leach easily into the environment following changes in temperature, pressure, or pH, or following irradiation exposure such as UV, microwave, and sunlight. So how readily these phthalates leach into the environment is largely dependent on the length of the alkali chain, with more longer length phthalates leaching having slower migration times within the plastic matrix and longer retention times. Similarly, shorter alkali chain phthalates leach into the environment much easier. These chemicals have short half-lives. However, due to their ongoing release into the environment, they can cause chronic exposure effects similar to the accumulative effects of persistent organic pollutants. 4 million tons of phthalates are actually produced annually on the global scale. And aside from a direct leachage into the marine environment from micro and macroplastics, they can also be transported into the oceans through air pollution, through air, soil, wastewater and effluent, rainwater, surface water runoff, sewage sludge, sediment, and stormwater. Once in the body, they undergo two phases of metabolism. In the first phase, they are hydrolyzed by esterase and lipase in the intestine parenchyma. In the intestine parenchyma, this increases their hydrophilicness. In the second phase, they are then biotransformed into the sulfo and glucoconjugates, turning them into their primary metabolites. Sometimes they undergo a third phase of metabolism where they oxidize into their secondary metabolites. So these phthalates undergo impart their toxic effects by acting as endocrine disrupting chemicals. So these can interfere with the endocrine system by acting as hormone sensitizers and target nuclear receptors and other transcriptional factors related to the endocrine system, such as the peroxisome proliferator activated receptors, referred here as PPAR, thyroid hormone receptors, and estrogen hormone receptors. And through acting upon these nuclear receptors have been seen to cause cancer, oxidative damage, diabetes, obesity, and inhibit reproduction. So the effects of phthalates on reproduction is of particular concern in humpback whales, as they only reproduce once every 3 years. So they affect reproduction by inhibiting the production of estradiol and hormones involved in pubertal development and ovary function. They can also disrupt the expression of genes involved in cholesterol transport and steroidogenesis, causing reproductive defects, disrupting oocyte maturation and embryonic development. They can also affect, however, their effect on reproduction is not limited to females, as they also can cause increased semen quality and testicular dysgenesis syndrome in males. This was seen to be dose dependent on urinary phthalate concentrations. So though humpback whales are a very charismatic species in of themselves, they also have various ecological roles within their environment. They have been shown to fertilize the oceans by recycling vital macro and micronutrients, the most prominent of which is iron. Now, iron is the second largest limiting factor in primary production, with both organic and inorganic fertilization experiments showing a 100-fold increase in primary production in Southern Ocean surface waters. Additionally, humpback whales help facilitate primary production via their vertical migration, which helps transport vital nutrients from the more nutrient-rich surface waters to the more nutrient-devoid deeper layers of the ocean, helping not only keep ecosystems healthy, but also support human food security. Now, although humpback whale populations have experienced incredible so although humpback whale populations have increased since the cessation of whaling in the 20th century, due to increases in both globalization and consumerism, other threats have become more prevalent, such as ship strike, noise pollution, climate change, and persistent organic pollutants. Additionally, the feeding method of humpback whales, so they're lunge feeders, which means they intake large volumes of water at once, and the extreme energetic demand of humpback whales, so they have I think it is the second longest migration of any species. These in combination put them at an increased risk of phthalate exposure. So as it stands today, there is very limited research on both the exposure and effects of phthalates on various marine mammals. However, there has been research on rhesus dolphin, the bottlenose dolphin, striped dolphin, harbour porpoise, fin whale, blue, and bowhead whales. There is only the one study looking at the effects of phthalates on marine mammals, with DHP being exposed to fin whale cells and being found to cause an increase in thyroid hormone beta-mediated luciferase activity by 22 to 27%. Now, this was observed in using 25 and 50 micromolar concentrations, and though these are pretty high concentrations, concentrations even exceeding this have been observed in the Mediterranean Sea, so it is still environmentally relevant. As it stands today, there are no studies on humpback whales in particular, despite their global distribution and their extreme migration, putting them at higher risk. So this study sought to compare the phthalate burdens of two populations, one in the that migrates through a relatively clean path, the E1 population. Now, this population migrates from their feeding ground in Antarctica, from their breeding ground to their breeding ground in the Great Barrier Reef. And our other population that we're looking at is the NA population. Now, this population migrates through the North Atlantic garbage patch off the coast of Bermuda. So the E1 population was sampled off the southeast coast of Queensland, Australia, off North Stradbroke Island, and the North Atlantic population off the coast of Bermuda through the garbage patch. Now, 6 samples were taken from each population from 6 different individuals, and these were skin and blubber samples taken from using biopsy darts and a modified air rifle for the NA and E1 populations, respectively. Both of these populations were sampled in their respective 2014 northern migrations, and 8 target phthalates were analysed. Now, this analysis was done in a specialised clean lab to prevent cross-contamination from airborne phthalates. Now, once the skin and blubber were separated and weighed, the blubber was then homogenized before being spiked with a surrogate standard. This was then a mixture of dichloromethane and hexane was then added before it was shaken and extracts performed in an ultrasonic bath. This was then centrifuged. So this process was repeated 3 times, and the resulting extracts combined together before being reduced down in volume using a stream of ultra-pure nitrogen. Sulfuric acid was then added and the extract was vortexed. This was then let stand for 48 hours before being centrifuged. At this stage, the organic phase was collected and the acidic phase was then washed with hexane. These 2 phases could then be combined together before being further reduced in volume and run through a sodium sulfate column. This was then reduced in volume 1 more time before gas chromatography mass spectrometry analysis could occur, giving us the calibration curves that were used to quantify the 8 target phthalates. Now, only 3 phthalates were detected in concentrations that exceeded their respective limits of detection. These were DEHP, DIBP, and DEP. However, DEP was only found in concentrations over their limits of detection in the 1E1 sample and therefore was not included in comparisons. DEHP was detected at a wide range of concentrations and had a higher standard deviation than DIBP, and DEHP had a high detection rate in the E1 population, whereas detection rate was the same in the North Atlantic population. So, as is expected, DEHP was found in higher concentrations than DIBP in both populations. Now, this was expected as similar results have been seen throughout the literature, including other studies looking at marine mammals in particular. What was also expected was the increased DEHP in the North Atlantic population, so our dirty population, compared to the E1, so our relatively clean population. This is likely not only due to the garbage patch, but also due to the sample site's proximity to Bermuda. Well, Bermuda has the highest density of humans anywhere in the world, and also has very poor sewage. And as mentioned before, both treated and untreated sewage has been observed to be a source of phthalate contamination into the environment. What was not expected was the increase of DIBP in the E1 population compared to what we understand to be the dirtier environment, the North Atlantic population. Now, this could just be a result of the limited sample size, as only 6 samples were used per population. However, DIBP is the predominant phthalate used in PVC manufacturing in Australia, so this could explain this result. However, due to the limits of the study, this cannot be said for sure. So as mentioned, there is very limited research on the instances of phthalate exposure and their effects on marine mammals. So further research should concentrate on not only the target phthalates analyzed here, but their primary metabolites, as these phthalates are metabolized so quickly within the body. Looking at the primary metabolites may give a more holistic view of phthalate exposure. Additionally, both phthalates and the metabolites should be investigated on their effects on transcriptional factors, aside from thyroid hormone receptors such as PPAR and estrogen hormone receptors. Lastly, their effects on energy provisioning, so their effects on mitochondria, should be investigated due to the potential for phthalates to potentially impact their migration and therefore reproduction of humpback whales. Lastly, the phthalate and plastic concentrations of both of these environments should be found, as it was only assumed that there were higher plastic concentrations and microplastic concentrations in the garbage patch than there was off the coast of North Stradbroke Island. So finding this will validate this assumption and give more weight to the findings. Thank you.
Thank you very much, Lara, for that presentation. Do we have questions for Lara from the audience?
Oh, there's no questions in the box, but if I may ask a question, Lara, if that's okay. It was around the estimates, your body burden estimates and the error. There weren't error bars on that. I'm just wondering if you can comment what sort of sample size would be necessary. Do you think you had enough samples in there, enough individual whales to detect those differences? And what was that variation between individuals? What did that look like?
So there was a pretty high variation in the North Atlantic population. So we did only have 6 samples for each, but that was mainly due to the price of running this experiment. It's very expensive. So yeah, we didn't have a very large sample size. Yeah.
What would be your recommendation then on sort of an optimal sample size if money wasn't an option, and to really detect those differences and to reduce your error for averages?
So I would probably maybe about 25, but in addition to that, in a perfect world, I think it would be good to have like a yearly, look at the yearly variation between in these concentrations.
Okay, so you think it changes? It changes through time seasonally?
I just say that mainly because in the last 5 to 10 years, there have been differences in regulation of these phthalates. So DIPP, I think in the last 2 years, there has been changes in regulation in specifically the EU. So as different countries take up similar regulations, I think it'll be interesting to see how that changes.
Okay. Can I ask one more question?
Yeah.
But they're bioaccumulative. So is there capacity for the whales to remove these from their system once they're in? Or is it an ongoing accumulative burden that they carry through their lifetime?
Yeah, so with the phthalates, because they metabolize so quickly, they don't actually bioaccumulate. Okay. So I'm not sure if I was clear about it before, but what I meant by the chronic exposure risk is just because the phthalates are pumped into the environment at such a steady rate that they almost mimic the effects of POPs, but they are, they don't accumulate within the body in the same way.
Okay, sorry, I think I missed that. Thanks.
That's good.
I have a question too, Lara, and it's a little bit unfair of your supervisor asking a question, but I think it's really useful for this discussion as well. We targeted blubber obviously. Is there a better matrix? Obviously, we targeted blubber because that's the tissue we can get from living whales, but is there a better tissue for these compounds that you would advocate if you were biomonitoring in a different species?
Yeah. So the most common is probably blood and urine. These are good because, like I said, they metabolize really quickly. So a lot of these actually exit the body before they can actually be stored in the lipids of the animal. So yeah.
So what we're seeing in these blubber samples are likely to be a very small residue compared to what's circulating in the whales.
Mm-hmm.
Okay, do we have any other questions for Lara? We're still running a little bit ahead of schedule. If not, well done. Yeah, you got another? Perfect.
Yeah, I'll just ask,
ahead,
because
yeah.
we've got time. I mean, you spoke a bit about that exposure and effects, and I know it's very hard to have that data, and one study is not very many, but how can you relate those phthalate burdens to actual effects or impacts on humpbacks in terms of populational effects? I guess we can see individual effects, but how are we going to see that? Is it really something that is going to impact on their reproductive output or the reproductive success, or their, what are you expect, or their life duration? What are those? I know they're ADCs, but what are those? How do you think you can relate that exposure to actual effects, not at a cellular level, but at a whole organism and a populational level?
Yeah. So like I said, we only have the one study on the actual cellular effects. So I think the first step would be increasing, looking at the effects on different transcriptional factors, specifically PPAR, because that is the one that's most directly impactful to reproduction, I believe. And secondly, like I said before, I think it's important to look at the effects, the exposure through time, so then we can pair that with different monitoring data on the populations that we're looking at. Obviously, because they're humpback whales, it's we can't put them in a tank and like expose them and see what happens. So I think that's where I would start with that one. Yeah.
Okay.
I'm thinking about your question too, Kath. I don't think I have a better answer, except it's probably not possible because with these large free-roaming wildlife, we're really seeing cumulative effects of everything. So so it's important to take that cumulative effects perspective to any wildlife, particularly the polar feeding wildlife, as we're discussing today, to have a look at what proportion of the risk pie chart you would attribute to chemicals versus krill disappearing. Is a little bit of a tricky one. But definitely lots more questions in this area and lots more work to be done. But thank you very much, Lara, for presenting that data. We're moving on now to microplastics, and I also encourage you again to go have a look at our e-posters where there are two more, one microplastic, one nanoplastic presentation, as well as a presentation on legacy pesticides as well as perchlorate. But for now, we're moving into microplastics, and we have a pre-recorded lecture from Alex Alves from the University of Canterbury outlining microplastics in Antarctic snow. But I do believe Alex is online for questions afterwards. So if we've got our IT team who could bring up Alex's presentation, that would be terrific.
Kia ora from Christchurch, New Zealand. My name's Alex and I'm from the University of Canterbury. And today I'll be talking to you about microplastics in Antarctic snow. So to start with, I just wanted to talk a little bit about plastics in general. So plastics pose a real global threat to our environment. So you can see on this image on the right, there is the obvious environmental pollution associated with plastics. There's also the impacts that come from plastics production using non-renewable resources and the associated health implications from those. Since the 1950s, we've produced over 8 billion tonnes of plastics, and every single year our production rates of plastics is increasing, even though we do recognise there are these negative implications associated. Every year at the moment, we're producing over 380 million tonnes of plastics, and of that, 50% is single-use plastics, which means we use it once and throw it away. So we really do have a very big throwaway culture currently, and it's very much almost a out of sight, out of mind approach for a lot of people. Unfortunately, plastics can break down and they do end up in our environment in all different forms. So that brings us into microplastics. Microplastics are synthetic polymers which are less than 5 millimeters in size. They can be both primary or secondary. Primary microplastics are those particles which are actually produced to be less than 5 millimeters in size during production. So it's things like your microbeads, which you might remember being found in your face scrubs or body scrubs in the past, and also things like pre-production plastic pellets, which is often how they produce virgin plastics, and then they use those to actually produce those larger plastic products. Secondary microplastics are those plastic particles which basically degrade or fragment or break down from larger plastic products through environmental stresses, such as UV degradation or wave friction and just general wear and tear. So things like washing your clothes as well can produce those secondary microplastics. In microplastic research, we commonly group microplastics into 4 main different morphotypes or shapes, and those are films, fibers, fragments, or beads. So we do this basically to categorize them, but also this helps us in further research looking how they move through the air, and then also it can help us look at potential sources. So fibers are usually associated with textiles and clothing. We know that microplastics are harmful to organisms from previous research and that they're found across the world in the air, water and soils. We know that plants are able to uptake microplastics and it can influence their growth rates, and we know that we're inhaling microplastics through the air on a daily basis. Recent research has found them in our blood and in our lungs, and so we know that we're exposed to these toxic chemicals associated with plastic production. Unfortunately, remote regions of the world are also not able to avoid the impact of microplastics. And this brings us into our area, so looking at microplastics in Antarctica. So previous studies have had a focus on the marine environment in Antarctica, so that being looking at sea ice cores, general seawater studies, and also things like marine sediment and benthic organisms. But there's limited data that exists on airborne microplastic concentrations in remote regions, particularly in the southern hemisphere, and there's also a lack of data on terrestrial microplastics in Antarctica. So our aim of this study was basically to further understand the extent of microplastic pollution in remote regions and the pathways into Antarctica. So to show you how we started, so back in 2019, I was fortunate enough to go down with the University of Canterbury Gateway Antarctica's postgraduate program. And as part of that, I collected these snow samples. So we collected fresh snow samples from surface snow, and we collected these from 19 sites across the Ross Island region. So as you can see here, the bases are marked with the blue little triangles, and so we collected some from closest to those bases and also some further afield so we could look at that distribution. So once I brought these samples back to New Zealand, I was able to prep them in the lab for analysis. This basically included doing a wet peroxide oxidation digestion, which just eliminates as much organic material from those snow samples as possible, so it's easier to look at those down the track. Once I had filtered those onto glass fiber filters, I was able to use a stereo microscope to analyze and categorize those particles that we were thinking could be plastics, and so I would look at their color, shape and size. Once I had found these, I used micro-Fourier transform infrared spectroscopy to actually analyze these particles, which can confirm the polymer composition of those. And as we were looking at fresh snow from either during a snowfall event or close to or reasonably soon after, we wanted to look at where those potential sources could have come from through snowfall. So we used high split back trajectory modelling to do this. So what do we find? So we found that microplastics were present at all sites that we sampled from. So those 19 samples we took, there was microplastics in all of those. We found them at an average concentration of 29 particles per litre of melted snow across all of those sites. And the highest levels we were looking at were around 84 particles per litre, so quite significantly higher. We found 13 different types of polymers across these samples, and 41% of those were PET or polyethylene terephthalate, which helps us link it to those sources as well at the end. And the highest proportion of particles we found were in the size range of less than 200 microns. So basically, the smaller we got, the more we found. Our analysis method is limited to about 50 microns due to the manual process involved. And so if we were able to use different analytical techniques, we may be able to see that there are more particles in that micro-nano range, less than 50 microns as well. And as you can see in this figure on the right, the larger the bubbles, the higher the concentration of microplastics at those sites. And we did see that there was higher concentrations of microplastics at those sites closer to the bases. We also found that there was higher concentrations in our samples than the surrounding marine concentrations from the Ross Sea, which is quite an interesting aspect to look at. So then we looked at the possible sources. And so we wanted to look at both those local sources and the long-range transport routes that may have gotten these particles into these samples which we collected. So on the left are some examples of the local sources. So there's obviously all of the building and equipment and bases down there in this region we were sampling from. And so everything from paint to equipment usually is polymer based. So this is a potential source. We also have these remote field camps that go and set up in these remoter areas as well, or further out at field. And so anytime there is a camp set up, there's likely to be microplastics left in the environment just from our human footprint down there. Another interesting one was the marker flags. We did find some microplastics in our samples that were nylon or polyamide, and they looked quite similar to the pink colour of these marker flags. And these are commonly used down to mark safe routes in Antarctica, so incredibly important for health and safety. But they're placed every 30 metres. So as you can imagine, there is a lot of flags made out of this nylon material down there. And this image just shows how degraded they become after being left out on the ice for a season. The Antarctic environment is obviously quite harsh, so all of these break down and are potentially ending up in these samples as well. And the other thing that we went and looked at in more detail was we catalogued the field gear provided by the New Zealand Antarctic program and looked at the polymer composition of the gear. Most of it is plastic based, which obviously just withstands the environment down there and provides the warmth you need. But it did mean that we had some links to those PET particles we were finding through looking at the gear. Then we looked at the long-range transport through high-split back trajectories, which we were talking about before. And as you can see from these images on the right, those sources look mostly like they're coming from the Southern Ocean. So they came from over 6,000 kilometres away in some of these cases, which means there is potential that they were coming out of the Southern Ocean, being picked up, and then transported and deposited through that snowfall event, which we then sampled from. We know that there is a marine to air exchange of microplastics, so this is a potential source. And there's obviously other sites along the way that they may have travelled past that pick those up. But there is this potential source of them coming from a very long route to where we then sampled from. So what does this all mean? So it really highlights an emerging pollutant of concern in the Antarctic. Airborne transport of microplastics enables them to reach remote regions, and microplastics, we know they can increase pressure on ecosystems, so their presence in the Antarctic is a major concern. We also know that airborne microplastics may contribute to climate change and accelerate melting of snow and ice, which is a growing research area at the moment. And so that's a really important thing to keep in mind for those wider implications. And lastly, it identifies the need for urgency in creating successful policy to reduce the extent and effects of microplastics across the world. So thank you very much for listening, and thank you to those who have helped make this work possible. Please feel free to contact me on the following email with any questions, and thank you to our funding and logistics providers as well. Thank you.
Well, what a great presentation. I don't think Alex is online. I expected her to be. Alex, if you are, maybe just jump in. Okay, I don't think she is, unfortunately. There's been some great work surrounding, great promotion surrounding this work as well. And I certainly had some questions about that work. I was keen to discuss it, but we all saw the email address where we can contact Alex if there are any questions. That means that wraps up our seminar this morning, the formal presentation aspects of the session. We do have some questions in the chat.
Yes.
And the first one, Andrea, is for you, so I'll ask it. It's the first two questions are from Javier Moreno, and thank you for joining us this morning, Javier. Andrea, what do you expect the concentrations of PFAS to be from other parts of the Antarctic Ocean?
Thank you, Javier. I hope to pronounce correctly your name for the question. It's hard to say now what we expect for the future, the concentration of PFAS in the Antarctic oceans, because of their slow path across the current oceans. Now the concentration are relatively low because of the Antarctic circumpolar current that protect the influence from the other currents outside the polar front. There are some concentration of contaminants, maybe because of the local sources, but probably from 2030, we will expect the peak of the concentration of these contaminants because of their usage in the 80s. It's hard to predict how much the concentration will be in the Antarctic oceans, but probably higher than now, than how can detect now in these years of starting research and trying to fulfill the gap of information in this remote area. I think it's difficult, Susan, to predict something for the future.
I agree. I'll expand on Javier's question a little bit, just for the sake of discussion. So I guess we looked at the East Antarctic sector,
yes,
and have plans of looking near the
Western
peninsula.
Antarctic sector.
Because of the way the Antarctic circumpolar current behaves in that region, where there is a physical forcing of the current across the peninsula. Would you expect those timelines to be disrupted possibly in that region?
I think because of the Western region and the Antarctica Peninsula here close to the South America, for example, maybe we can expect higher influence of the contaminants from a place that use this kind of products. So from cities and maybe from rivers and other sources near the Antarctica Peninsula. So in my opinion, maybe we could find in the transect from the South America to the Antarctica Peninsula a higher concentration of these contaminants. This is what I think, but we can't know for until we collect some samples.
Yeah, we can test that hypothesis. But yeah, I think Javier, I would agree, and also there would be a more confounded effect from atmospheric versus oceanic transport due to the distance being much shorter. But certainly an area that is, I guess, as Andrea, you concluded in your presentation, it's very simple sampling, and there is a need for capturing this timeline. So
exactly.
I'm sorry, may I ask to Andrea also?
Yeah,
of course.
Yes, Andrea, it's very beautiful presentation and very useful. And my question is any measures of PFAS or P4A in wastewater formed at Antarctic stations, like PBDs measures or HBCD hexabromоcyclododecan. Both of these compounds were measured and were found in wastewater, and thus it was identified the local sources of these new POPs in Antarctica. And what about P4S?
In the western sector near to the Antarctica Peninsula, yes, previous studies identified some of these contaminants, probably from the research stations. But despite the Arctic environment that it's full of papers and research about these contaminants, the Antarctica continent is still in lack of information. So there are some results and some identification of these contaminants, but are at the moment very low amount of data. That's why I'm programming to go and collect other samples to try to fulfill this gap that is really annoying, how much lack of information we have about this environment so fragile. And that's, yes, there are evidence of contamination, but we need to improve our knowledge because it's really poor in this moment.
I'll jump in, Tamara. We certainly have data from also the Australian station in wastewater and dust,
Yes.
and also transects away from the stations, and I think we can use those stations as you did in your study as proxies for other stations, because as the global community, we are using these consumable products containing these chemicals. And as Andrea presented, there were peaks closer to the station that certainly elicit the thought that these are local emissions rather than long-range transport, and that's an interesting finding, because the original hypothesis, of course, was we wouldn't find them south of the Antarctic circumpolar current, but we clearly did, and so local sources do need to be considered.
Thank you very much.
Andrea, do you want to take over reading of the remaining questions?
Yeah, of course. The second question is for Lara. from Javier again. He questioned, when looking at these results, would you say there is an urgent need to take measures of phthalate liberation to the ocean?
So I would say yes, especially with what we know of their potential effects on other species, such as humans and rodents. That's what the majority of the research is on. However, we do need probably a lot more research on marine mammals in particular in order to push the regulation that we would need. But I think it would be actually be a more meaningful step if we almost changed the system so we can actually test these chemicals on their effect on the environment before they are used commercially, because usually what happens is we will have increased regulation for a particular chemical and then we'll just replace it with a new one, and then 5-10 years later we realize that it's just as bad, if not worse, than these ones. So yeah.
Yes, unfortunately so. That's a good response there, Lara, and also I guess the most powerful data always comes from effects. So adding to that effect data would really add to our evidence for regulation as well. Do we have any broad questions now? We seem to have exhausted our individual questions per presenters, but we did want to provide this platform for this community today for some broader discussion as well. We've waited a long time for this conference to take place during COVID and so forth, and so I deliberately don't want to adhere to any strict format here. I simply want to create the platform for any discussions in this area that anyone now simply wants to bring up, something that they think is important in the field, something a lot of us are involved in the various SCAR action groups surrounding contaminants. So if there are any thoughts that people want to raise, any discussion items branching out from the presentations or indeed the e-posters, then by all means jump online. I can see one hand up. Yes, Kath.
Catherine.
Thanks. Yeah, it's more to open up a bit of a discussion about the question. It's been raised a couple of times now, the question of proportion of microplastics or other contaminants that you'd attribute to local sources versus long range transported sources, whether that's airborne or oceanic. And I think as a community, this is something really that we need to address urgently in mind and address in terms of our operational objectives and the measures that we put through our operational stations in Antarctica. Because if we can prove that local sources are significant in terms of accumulation and impacts, then we're better able to not pressure, that's probably the wrong word, to influence our activities on station and the products we use and the methods we use for treatment and all the rest of it. So it's really more a discussion point, but if anyone has some information that they'd like to share on that proportional that you could attribute to local versus contaminated sources, I'd be really interested to hear. I know there's not much data, but yeah, if there's anything that people want to contribute on that, I'd be interested to hear that.
It's really interesting and very timely, Kath. I know both Nicoletta and I and others are involved in the AMAP local versus remote sources expert group at the moment, and it's of course in the Arctic there are many more sources because of the human populations that live all year round there, but I very much agree with your sentiment that when we are discussing contaminants, particularly organic contaminants in Antarctica, it becomes such a big whose problem is it? Because we're talking about hemispheric problems. Whereas the age we're moving into now is that the chemicals aren't diffuse, hemispheric, persistent, semi-volatile or hydrophilic contaminants so much as continuous emissions of everyday consumer chemicals such as the phthalates or such as other pharmaceuticals and so forth. And if we can help, for example, COMNAP, which would be the body in Antarctica, to elucidate some products that we now know are not so great. I think that would be a great step. So Tamara and Sergi's work on the practices that we undertake in Antarctica and further work on products such as, well, I guess the obvious one is a PFAS being used in so many different applications that are great in Antarctica, such as firefighting and waterproofing. And then we may be able to better advise them of what product alternatives are better. And that's the kind of practical advice I think, Kath, you're also alluding to, that would be really useful to say, look, this is a problem that we play a role in that, and therefore we can play a role in mitigating, rather than putting it all in the global basket.
Yeah, and I think to affect change on the ground at our stations, in our operations, is it's going to have to be evidence-based. We're going to have to provide scientific data to say that it is causing a problem and we need to do something to change our operations to reduce it or to remediate that.
Yeah, absolutely.
It's just something that we can control, I think. It's just it's a lack of data, really, that nations won't respond unless there's evidence that they're doing some harm. They won't be forced to respond. So
yeah. Absolutely. Well, maybe we roll Antarctica into the assessment that's currently going on. There's certainly scope to do that, if there is enough interest from the Antarctic governance community to roll that in. That could be done hand in hand with AMAP. Any other questions from the group here today, or discussion items, priming questions?
Just while people are having a bit of a think about that, Susan, I just wanted to mention that there's another session tomorrow in the Solutions to Pollution, which is contaminant impacts and remediation. And the session tomorrow is particularly focused around microplastics and nanoplastics. So if people are interested in that and it's not at some ungodly hour, that's on at 0300 UTC time. So I don't know what that means for other people, but it's a few hours back for me. So yeah, but if people are interested, that one's happening tomorrow, and there's some interesting talks on microplastics.
That's excellent. It's great to see so much work coming out for the region in the plastics area. So that's excellent. I'll just read out our, perhaps whilst people are having a think as well, our e-posters are perchlorate occurrence at different points in Antarctica during 2017 to 2020 by Rosa Azevedo Barrios. Number 2 poster is toxic anthropogenic signatures of pesticides in the superficial sediments of Pritz Bay, East Antarctica, by Ashwati Shaji. Thirdly, assessing single and combined exposures to nanoplastics and nanometals in oxygen consumption, lipid peroxidation, and protein carbonylation in Laternula elliptica by Rodolfo Rondon. And finally, occurrence and characterization of microplastic particles from carcasses of Adelie penguins collected from the Antarctic region by Sharan Bhattacharjee. And I apologize for any incorrect pronunciation. Okay, how are we going? Is there any other appetite for discussion, or have we drained the conversation on this platform this morning? It doesn't look like we've got anyone else jumping in. So I would like to sort of formally close the session, and thank you for you very much to our presenters and to our participants, and I would
sorry, Susan, there
yes?
is another question from Javier.
Yes.
When working with such low concentration of analyte over such a large area, as it the ocean or the Antarctic continent or the atmosphere, is there any concern to the accuracy of the results in these measurements? Yeah. This is a interesting question. I don't know. It's not for me. I think it's a general question about us that we gonna try to detect some chemical compounds in this large environment.
I think it's a problem we've analytical chemists have always faced, Javier, and the issue is magnified in Antarctica, where we often have to extract very large volumes of media to attain accuracy. And when we do that, of course, we're also increasing the natural background interference. The important thing, I think, here are our quality assurance measures when we're doing this work. So this is actually a task list the impact group is working with at the moment to develop those quality assurance criteria and ensure that that all publications have comparable quality assurance criteria. So if we can say that we are detecting contaminants above blank levels, above instrument noise levels, if we're using isotope labeled standards, recovery standards, then we can trust our data. But that the level of our confidence in our data is, of course, related directly to the stringency of our quality assurance measures. And it's important that they're in place when we're dealing with an environment where often, particularly with new compounds, the methods are under continuous improvement, and we are pushing the levels of detection. So it's very important that they are in place, and that's not new to Antarctic. That's not something unique to Antarctic analytical chemistry, but it is, detection is often pushed at the boundaries in Antarctica. So it's a really important issue that you've opened up there. And I think I'll pause in case someone else wants to share the woes of analytical chemistry in the Antarctic context, or jump in with any other comments there. I did see that there was another question for you, Andrea, from Jiangong O as well, that he would like to discuss further with you by email. So that's excellent. But yes. I will remain online as well for any further discussion. That was simply a formal sort of close to the session in case other people want to get on with their day or their evening. But I'll certainly hang around here, and I'm keen to chat and discuss Antarctic chemical pollution research with anyone interested. Otherwise, have a very good rest of your day.
Catherine, your microphone is mute.
Still haven't got that right. Thank you, Susan, and thank you all for a really interesting session. I enjoyed all those talks. I'm going to head off home because I've been online now for 6 hours, and it's not that late here, but I'll leave you to it. But thank you very much. It was a great session. All right. Bye, guys.
Thanks for your help, Kat.
See ya.
Take care.
Thanks to you.
Bye.
Hi to everyone.
Thank you very much. Bye.
Thank you, Tamara.
Thank you. Thank you so much.
Thank you, Susan. Thank you, Andrea. Bye-bye.
Thanks.
Bye bye.
Thank you.
Thank you.
Sir, should we close the session?
Yes, if we've only got 2 participants left. Yes, thank you.